everyone. I'll be presenting to you today my project on the characterization of a therapeutic approach to deliver scFvs, these are fragments of antibodies, single-chain variable fragments, to target TDP-43 proteinopathy in ALS. So our project is divided into three big parts, and I'm going to go through each one of them briefly with you today. So the first part of the project was about identifying scFvs that can bind to TDP-43, and that was done through a phage display screening, where we identified two scFvs that I'm going to call B1 and D7 targeting TDP-43. Then we confirmed their binding and we checked their effect in cellular models of ALS. So first of all, to confirm their binding, we did an ELISA, and as you can see here, we do have binding between B1 and D7 and not the control to wild-type TDP-43. Then we did in silico binding site prediction to see where the scFvs can potentially bind to TDP-43, and we saw that they can probably bind to two pathologically relevant domains such as the RRM1 and the C-terminal domain of TDP-43. Then we wanted to test the effect of these scFvs in cellular models of ALS, so we used two models. We used HAC293 T cells and the motoneuron-like cells, the NSC34, overexpressing wild-type TDP-43. So when we express the scFv G7 in these cells, we see that it causes a decrease in the fragment, in the insoluble amount of the 35 kilodalton fragment of TDP-43. Then when we inhibit the proteasome, we see that this effect is abolished, so we think that it is mediated by proteasomal degradation of this fragment. And when we express the scFv B1 in cells, we see that it causes a decrease in the activation of NF-kappa B, which was caused by the overexpression of TDP-43. Now, the second part of our project was about enhancing the delivery of the scFv in its protein form to target cells, which will then be applied also to mice in order to help us cross the blood-brain barrier. So in order to do so, we decided to use nanoparticles that are known as spions. Now, spions are nanoparticles that are only magnetic in the presence of an external magnetic field. And what's interesting about these particles is that they have theragnostic properties. So this means that they can be used for therapeutic purposes, so to deliver therapies to target cells, but they can also be used for diagnostic purposes because they can be tracked inside the body using MRI. So we tested different formulations of scFvs and spions with different mass ratios and amounts of spions to scFvs. And we tested them for their size, for their surface charge, and for their ability to retain the scFv. So we saw that their hydrodynamic diameter was less than 200 nanometers, which is the baseline for crossing, or the threshold for crossing the cellular membrane. And we saw that they have a neutral zeta potential, so two parameters that are compatible with cellular internalization. Then we did something called the heparin decomplexation assay to confirm that indeed our scFv is complex to our spion. So basically, here we can only detect on the blot the free scFv that is not complex to the nanoparticle. And when we add the spions, we see that we detect less free scFv, so that was good. And then to confirm that it is actually bound to the spion, we add heparin, which will compete with the scFv, and it will decomplex it from the nanoparticle, and then we detect it again on the blood. So this was just a confirmation that the nanoparticles are well complexed. Then we did some toxicity assays on cells to try to determine the concentrations and the time of treatment that would not be toxic to cells. So again, we tried different concentrations and formulations and different times of treatment. So after 4 hours of treatment, we saw no toxicity whatsoever on HEC293 T cells with any of the concentrations that were used. But at 24 hours of treatment, when we go into the higher concentrations, we start to see some toxicity. So because of that, we decided to stick to the 4 hours treatment. And after 4 hours, we wanted to check if our scFv is internalized into our cells. And as you can see here, with the flow cytometry, we're able to detect the signal coming from the spion. We have the shift in the signal, so it shows that the spion is well internalized into the cells. And by Western blot, we were able to detect the scFv that was also internalized into the cells when we added the spions. Now, what was interesting here is that in the presence of spions, we detected a 30-fold increase in the amount of scFv that was internalized into cells compared to when the scFv was delivered using a normal protein transfection reagent. So that was a pretty interesting result that we got. And the third part of our project, which was running in parallel to the first two parts, was the full characterization of an animal model of ALS with a TDP43 mutation. And basically, we are trying to look for parameters that haven't really been tested before in this specific model. So we decided to use the TDP43 model with the mutation A315T. And first of all, we saw that the onset of symptoms was around 150 days of age, after which we saw that there is a 44% difference in body weight and a 45% decrease in grip strength. We also did some other tests like tail position and gait impairment, and all of those also showed an onset around 150 days. We also did some MRI studies to try to detect structural changes at the level of the central nervous system, more particularly at the level of the spinal cord. So we chose to do that at 3 months, 6 months, and 9 months of age. Now, at 3 months, we saw basically no change. At 6 months, we start to see some change, but it was not significant. And now we are waiting for December to do the 9-month-old mice to see if we detect a change then. And the point is to try to correlate what's happening at the level of the structure of the spinal cord with other parameters that we are measuring during disease progression. Then we tested the expression of different proteins in different organs. I'm going to present to you here just the brain and the spinal cord tissues. So obviously we started with TDP43, and we have an increase in the total amount of TDP43 and its 35 and 25 kilodalton fragments at the level of the brain. At the level of the spinal cord, we saw the same thing, but we weren't able to detect the TDP25 fragments. Then we also saw an increase in the amount of the insoluble TDP43 and TDP35, and in the amount of phosphorylated TDP43. We tested also some other proteins that are related to ALS, and we saw that there was an increase in the GFAP signal, so a protein, it's a marker of astrogliosis. At the level of the brain, we saw an increase in the expression of NRF2, which is a transcription factor related to the cellular response to oxidative stress. And we saw a decrease in the ratio of phospho-AMPK to AMPK, which is related to the response of the cell to its bioenergetic status, so to metabolic stress, for example. Now, these changes were not significant in the spinal cord. We also tested some other proteins, some autophagy markers, the IBA1 marker, and we saw no change in those either. Now, a very interesting result that we got is when we did the metabolomics analysis on dry blood spots taken from before, at symptom onset, and following symptom onset on mice. And here I show you the result that was that we got recently upon the onset of symptoms. And as you can see, we have a very clear separation of the metabolomic profiles between the wild type and the transgenic mice. And the metabolites that were mostly changed were related to phospholipids, to some amino acids, to creatinine. So we need to analyze these results more. But what's important here is to say that metabolomic changes, they seem to happen very early in the disease, and they seem to be a good model to separate transgenic from control mice. So that's it. To conclude, we were able to identify 2 scFvs that can bind to TDP43, wild-type TDP43, and target its proteinopathy in ALS. We were able also to enhance the internalization of these scFvs into target cells using nanoparticles called spions. And we are characterizing more deeply the TDP43 mouse model, and we saw an important result that was related to metabolomics that showed a clear difference in the profiles between transgenic and control mice. So future experiments, we're gonna work on characterizing even more the binding between the scFv and TDP43, and we're gonna do that using TDP43 mutants and surface plasmon resonance to determine the exact binding sites and the affinity. We're gonna also work on continuing the MRI, but we're also gonna do some further metabolomics, transcriptomics, and targeted proteomics. Another thing we're doing is comparing the microbiome profile between transgenic and control mice to see if there is any correlation with the disease. We are also working on the spion scFv complex approach in mice, and at the same time we're developing some adeno-associated viral vector delivery system for the scFv into the central nervous system to finally study the effect of this scFv on the progression of symptoms in mice. So I would like to thank everyone who was implicated in this project, and I would like to thank more particularly the ARSLA for funding my 4th PhD year, which I'm going to start soon. And I thank you all for your attention.
Thank you very much. A question.
Hi, thanks for your presentation. I have 2 small questions and one very naive on the spion. How do they get, and how long do they stay in the body or the cell? How do they get degraded? So how long would that stay in? And my second question is, when you showed the data about TDP43, basically the full one and the 3525, I assume it's not what you were expecting, no? Or were you expecting it to go up? It
did go up. So I'll answer the second question first. It did actually go up, which was something we expected. So we have an increased fragmentation of TDP43, we have increased aggregation of TDP43, and the total amount of the protein was also increased. Given that TDP43 also autoregulates and all, so this was kind of expected. We expected that result.
But not with the spion, we agree? If you would put you would expect that it goes down, no? Or I missed something?
Here it was not treated.
Okay.
It was so just the mice, untreated mice.
I see, sorry.
Yeah.
And for the spion, the degradation, how long could you
It changes
target
a lot. It changes a lot depending on the coating of the spions. So we can also actually increase or decrease its half-life in the body according to how we coat it. So this is something we're still working on. Now it's only coated with the PEG. But it is part of it can be excreted by the kidneys. Part of it is metabolized inside the cells and they're degraded. Some of them are also apparently metabolized at the level of the lungs, and they change a lot according to the size of the particle and the coating. So this is something to be determined later.
Thank you.